I don't know what the collective noun for a group of computer screens are, but I would just quite like to welcome you to our NSA and Moredun shared webinar about ticks and problems with ticks and sheep. And we have two very eminent experts that are going to speak to us tonight. I am one of the two Scottish trustees on the board of NSA, and I've been working with gamekeepers for the last 10 years, working on tick control to try and reduce the tick incidence in the grouse on the Heather Hill here in Glenesk in the east of Scotland. And today's meeting is going to be recorded, so if you miss anything, you'll be able to rewind and listen to it again. It will be available after the end of today's meeting, and we'll have a question and answers at the end. And I think we're going to have a very lively chat today. So I'm looking forward to it immensely. Our first speaker today is Mara Rocchi. Mara graduated as a vet at Milano University, Italy, followed by and conducted a PhD in prion biology at the same university. She then went on to work on ruminant immunology at an international research institute in Nairobi, Kenya. In February 2000, she started working for the Moredun Foundation, focusing on immunology and vaccinology research in sheep and cattle. In 2014, she became head of Moredun's virus surveillance unit, which contributes to the development and deployment of tests for diagnosis of endemic diseases surveillance for the Scottish Government. She was involved in the development of the diagnostic test used for tick-borne fever, beginning her career in ticks and tick-borne diseases. In 2016, she worked in collaboration with Professor Lucy Gilbert, Glasgow University, advancing her career in the tick research area. Since 2016, the virus surveillance unit was developed a number of tests for the detection of tick-borne pathogens and ticks in livestock. She joins us tonight to discuss tick biology, tick surveillance, and tick-borne diseases, and I'm very much looking forward to Mara's presentation. Welcome you, everybody. Here's Mara. Thank you.
Thank you. Good evening. I hope you can hear me. I do apologize for my voice. I'm just recovering from the usual virus pestilence. But anyway, I'll do my best to be as clear as possible. I'm going to share my screen and we start directly. We're talking about tick and tick-borne diseases. And I have tailored this presentation to the UK situation. Great. Oops, sorry, this one is not moving. Okay, I need to stop share for a second and see if I can go back and get to the other talk. And this is not good. Sorry about that. Matt, are you still sharing your screen by any chance?
Here we go.
No, it's all yours.
I don't understand what is happening. Anyway, let's try this way.
That's it.
Right. Yeah, I want to see if I'm able to move between slides. Yes, I can. Okay. So we start with the very simple things. What are ticks and what do they look like? I'm pretty sure most of you have encountered ticks in their life. I just want to remind you that ticks are small arachnids. They are not insects, but also they do require a blood meal to complete their very complex life cycle. And there are over 800 species of ticks worldwide, but there are only 2 species, the Exodidae that are hard ticks and the Ergasidae that are soft ticks that can transmit disease. And if you can see here on the on the left-hand side, I'm sorry, I don't know if you can see my cursor, never mind. On the left-hand side on the top, you can see a questing hard ticks and an engorged hard ticks on the side. So it's a tick that has fed. Tick size changing depending on stage and sex, and the color varies depending on the species and they are fed or not. The reason why I'm telling you this is because if you encounter a tick that looks slightly different or it doesn't look right, I'm asking you please to collect it. Go online, look at the tick surveillance scheme, and send the ticks down to the UK Safety Agency. This is because there is an active program of surveillance to make sure that we can spot the early incursion of exotic ticks. Ixodes ricinus, the sheep ticks, is the most frequent species found in the UK on livestock, and the size of these particular ticks varies from a pinhead, and you can see it on the right-hand side of the screen, which is a larvae, 2 centimeters, that is a fed female. The blood meal. The blood meal is why we're actually here. Basically, without the blood meal and without the fact that the ticks can bite and transmit diseases, we won't have this seminar, we don't have this problem. Tick look for hosts by perching on low vegetation and waiting. This behavior is called questing, and on the first picture on the right-hand side, you can see a questing Ixodes ricinus female on a blade of grass. Although ticks cannot see and use the carbon dioxide and heat to detect prey, and to do that, they use a specific organ. If you look at the picture on the right-hand side, it's called the Haller organs. They have palpi that are like antennae that are sitting at the side of their head, and the Haller organ is at the top of those palpi. When a host passes by, the Haller organ detects the presence of CO2 and heat, and the tick jump from the blade of grass on top of the prey. Hard ticks search for hosts and feed during the daytime, and they return to the ground during the nighttime. And they live in woody, brushy area that provides food and cover for them and for hosts, such like mice and deer. How do they take the blood meal? To feed, the ticks insert this hypostome, that is a part of his mouthpiece, into the host skin, very similar to what a mosquito would do. And then you start to inject tick saliva that create a tick feeding cavity. The tick saliva contains cementing material that help the tick to stick to the host, but also contain a small amount of anesthetic, anticoagulant, immunosuppressive, and anti-inflammatory substances. All of those substances are facilitating the collection of the blood meal, but unfortunately the same substances help the pathogens that by now have moved in the salivary gland of the tick to infect the host. Ixodes ricinus is a representative example of ticks that undergo a 3 stages life cycle. The cycle starts with the eggs, which are laid by the gravid female. Over time, if the environmental conditions are favorable, larvae emerge from the eggs, climb the vegetation, and start to look for a host. Smaller the life stage of the tick, and smaller generally is the host. Therefore, host number 1 is generally represented by small rodents or small birds or other small mammals. After the blood meal, the fed larvae detach, fall to the ground, and over time they molt. When they molt, they change life stage and they transform into nymphs. Again, once the environmental conditions are favorable, the nymphs become active and start to quest for a second host. In this case, the second host are bigger mammals, like here, still some rats, rabbits, and some bigger birds. Again, once they're fed, the ticks detach, fall to the ground, take some time, molt, and become an adult. As you can see here, when the tick become adult, it's much easier to distinguish the female that usually are much bigger from the male. Both male and female climb on to their final host. Host number 3 generally is a deer, but could be a sheep, it could be a human, and they feed and mate on those hosts. After 5 to 15 days, the tick detach again, and the engorged female lay fall to the ground and lay eggs. Tick that don't feed become dormant, quiescent, and eventually die. And from the eggs here, we can restart the life cycle. So as you would have noticed that in the previous slide, I keep talking about the tick becoming active, because if the tick is quiescent or dormant, they will not jump on their host, therefore they will not cause a problem. But how do we measure the tick activity? Then tick activity normally is measured by number of ticks per drugs. So there is a person, an operator that drug a blanket, usually of white wool, over a specific area in a specific environment. At the end of the dragging, all the number of ticks that are attached to the blanket that are the active ticks are counted. And this will give you the number of ticks per square meter or square foot, whatever you like. And that's the number, the way in which we can compare different environment and different part of the, say, a field or a grassland to see which area contain the majority of the active ticks. So here I'll give you some relationship between tick activity and temperature that have been counted in this way, that have been analyzed in this way. As you can see, the temperature here, it goes from 5 degrees, from 15 degrees, and at 5 degrees, only less than 20% of your ticks are active. And then as you increase the temperature, there is almost an exponential increase in the activity of the ticks up to the point where you get between 11 and 12 degrees, the majority of the tick become active. Now, this data has been collected in Scotland. I am not so sure what, so we don't have data over 15 degrees, but what I would like to say is that if we went over to 20 to 25, we would start to see the activity decreasing because ticks don't like an environment that is too hot. Probably related to the temperature, we can look at tick activity and altitude. And you can see at 400 meter over the sea level, so there's approximately 12,000 feet, the majority of the ticks are active. And as soon as you start to increase with the altitude, the tick activity decreases. And this is probably because higher you go, especially in the UK, and lower the temperature here. Also, tick activity is related to habitat. If you compare an open habitat, like a grass moorland, with a woodland, you can see that approximately 5 more times active ticks are found in the woodland, probably because of the temperature, but also because of the humidity, because ticks, again, don't like an environment that is too dry. And putting all this data together, we can start to understand why we have those peak of activity at different time of the year. This is a figure that has been, I found on a Pennsylvania American website. So the seasons are not exactly representing what we see in the UK, but you can see that the 2 typical peaks of activity of adult ticks, spring, early summer, and early autumn, early and late autumn, are represented as well. And those are specific for adult ticks, where nymphs and larvae seems to be able to cope much better with the intermediate season that is the hot season. And then we move to tick surveillance. So there are a number of agency research groups that are conducting currently surveillance for ticks in the UK. The main one is the UK Health Security Agency. They run the tick surveillance scheme. This is for not only for Exodida, that are the one represented in this picture, but for all the ticks. And they have experts that can identify and classify the ticks that are submitted. You can Google it, and anyone from the public can submit ticks to the scheme. This is the data up to 2020 and show the distribution of Ixodes ricinus. You can see the south of England is mostly affected. There is another nice concentration in the north of England, and ticks are scattered basically all the way over Scotland. The Animal and Plant Health Agency ran a similar surveillance scheme. In this case, it's not for ticks, but tick-borne diseases. Is the APHA disease dashboard that is organized by species. I'm pretty sure most of you will be familiar. If not, have a look at it because it's a fantastic resource. In this case, I did look for tick-borne fever distribution in the UK. And as you can see, apart from this area where there is London and particularly larger number of species susceptible to ticks represented, tick-borne fever is present more or less, excuse me, in the whole UK. And this is a map that I have prepared, instead combining different resources regarding Lyme disease. And again, you can see that there is an overlap between the number of cases that are reported, excuse me, for Lyme disease and the tick distribution. Now we move to the causes of losses by ticks in the UK. There are direct causes of losses like Tick-Borne, blood loss, if you have a very high infestation, damage to the hides and light weight gain, or we can have specific diseases like Tick-Borne fever, male in cattle and sheep, louping gill, sheep, cattle and horse, babesia, that is water. And I will touch on Lyme disease because if you're exposed to livestock ticks, you might also be exposed to Lyme disease. Sorry, I managed to go in the wrong place. So we start with anaplasma, phagocytophilo, tick-borne fever or pasture fever in the cow. The disease is transmitted by Ixodes rhicinus, is caused by the bacteria Anaplasma phagocytophilum. Excuse me. You can see here in the white blood cells, and this is a cow that we recently diagnosed, these blue dots in the cytoplasm of the cells are the colony of bacteria. The disease affects ovine, but also bovine, equine, canine, and in some cases man. And the problem with anaplasma phagocytophilum is that it attacks the cells of the immune system. Therefore, they destroy the cells that are actually supposed to prevent the infection or to control the infection. The incubation is 5 to 15 days, often present as asymptomatic, but in some animals can cause high fever. And then the fever is followed by mild to severe immunosuppression. Due to the immunosuppression, we can have co-infection and secondary infection. And some of the secondary infection might be a bit strange, like mycotic pneumonia that is not particularly frequent. And that's probably the point in which your vet start to say, oh, maybe we have anaplasma, we have Tick-Borne fever on the farm. The high fever is also responsible for abortion storms in animals that have never encountered the disease before. If you have an abortion storm, please check the rams, because another consequence of anaplasma is that the rams can be infertile for a certain length of time. If you need to test as soon as possible and test several animals. The diagnosis is by blood smear, like the one that I showed you there, or through a molecular test that can be carried out on EDTA and on heparin blood. And here you can see a spleen from a normal lamb compared with the spleen from an animal with anaplasmosis. And this is a pneumonia that is a co-infection with Pasteurella and anaplasma. Louping gill is another tick-borne disease. As a tick-borne disease is widespread in the British Isles and predominantly affects sheep and red grouse, but the infection can also occur in other species, including man. It's often asymptomatic and may present as an acute fatal disease with neurological signs. If there is a tick-borne fever at the same time, the disease normally is much more aggressive. The causative agent is louping gill virus, and we have serological tests to do surveillance, screening, monitoring, but also diagnosis in live animal, and pathology or molecular tests to confirm disease, for example, in grouse that have been found dead on the move. The test can be used for a screening or for a diagnostic purposes. We have a vaccine in Moredun under development. And my suggestion, if you know that you are in an area where louping gill has been detected before, is to expose the lambs when still protected by the maternal colostrum. Here you can see those are cells in the cerebellum of the animal, and the brown dots, the brown stain, are all the cells that are infected by louping gill. And you can see one of the typical neurological presentation. And if you look at this map that has been produced by Franz Plalisaure from SRUC, you can see all the yellow dots are in Scotland, all the area in which louping gill have been detected. But as I said, sometimes can be asymptomatic, so you might have it in the flock and you might not know it. The third disease I want to touch on is babesiosis, red water. Babesia spp are parasites, so we had a bacteria, anaplasma, a virus, louping gill, and a parasite now, babesia, which infect the red blood cells. You can see on the right-hand side the red blood cells on a blood smear, and those things that look like two small drops or two small pairs are the babesia parasites multiplied inside the cells. The disease affects cattle, dog, human, horse, and sheep, goats, pigs, and sometimes cats. Some of the babesia are zoonotic. Unfortunately, some of the livestock babesia are zoonotic. Some of the other aren't. and they can be transmitted by a different number of tick species. The classical symptomatology with babesia is fever and hemoglobinuria, so you get dark urine, as you can see here in the bottom right-hand side. So if you have those symptoms, you know this is the time of the year, you can see that there is tick activity, you know that you had babesia previously on the farm. The best option is to try to contact your vet and see if a diagnosis can be obtained. There is inverse age immunity. Generally, younger animals are much more resistant than older animals to babesia. And the diagnostic can be carried out by smear, although it's quite difficult because you might have a very low number of cells with the parasite inside. It's very difficult to detect them, or by molecular methods. And Moredun, we are trying to develop a serological test. And here on this map, you can see where the babesia has been detected. That was up to 2018 in the UK. Finally, I want to touch on Lyme disease. Lyme disease is a human disease. Has been reported in other species, but the reports are not particularly conclusive. But the disease has been on the increase for the last few years. This has been accompanied, probably related with increase in number of ticks and the expansion of the area that the ticks are covering now. The disease is mainly caused by Borrelia burgdorferi or similar species of Borrelia. In this graph, you can see how between 1998 and 2011, the disease has continuously increased. And this is data from the Health Protection and Security Agency. And this is the Borrelia, the little spiral bacteria that you can see in the blue background. That's the cause of your disease. The disease mainly affects humans, and the ticks can transmit Borrelia anytime between 2 to 48 hours after bite. The diagnosis of the disease is challenging. Initially, you get flu-like symptoms. The majority of the people, approximately 70%, get a rash. This is a typical Borrelia rash. It's called the bullseye rash. So 2 concentric circles, they also increase in size and move. But remember, not everybody gets the rash. So if you have a tick on yourself, collect the tick and keep it or send it to the UK Health Security Agency. If you get a rash, take a picture and take the tick yourself and the picture of the rash to your GP. Generally, if the GP is working a disease in a Lyme disease area, it will be able to help you with the diagnosis. But there are some GP that are still not particularly familiar with the disease, so you might have to insist. And remember that you can always look for a second opinion if necessary. So I just wanted to leave you with a couple of resources. This seminar is going to be recorded, so you can go back to that one. This one is just telling you what to do and how to remove a tick from yourself. And the second is this list of useful resources that different agencies have produced, including Moredun, regarding tick-borne diseases and especially Lyme disease. Thank you.
Thank you very much, Mara. There's a lot of food for thought there, and I've certainly one or two questions I will ask you later on. But we'll move on to our second speaker tonight, Matt Colston. Matt is a veterinary graduate from Edinburgh University. He holds a RCVS, I don't know what that is, you'll have to tell me, Matthew, a certificate in sheep health and production. From 1986 to 2013, he worked in mixed general practice, mostly large animals. This included a 2-year teaching post in the vet school in Edinburgh. During this time, he served a term as president of the Sheep Veterinary Society and also spent 9 years as non-executive board member for the Moredun Foundation. Since 2013, he's been a veterinary technical consultant for Novartis and Elanco Animal Health. Matt joins us tonight to discuss tick control, including avoidance and management strategies. Matthew, if you could start, please.
Thank you, Peter. I'll just... share that. Can you see that one okay, Peter?
Yes.
Okay, thanks for the introduction, Peter, and thanks to Moredun and NSA for inviting me to speak this evening. There's nothing terribly groundbreaking about what I'm going to say this evening, so for you chaps that have been involved in tick control on the grouse moors It's the same old stuff, I'm afraid. And I've called this section Practicalities and Limitations, because although there is always something we can do to get better control of tick numbers and tick-borne diseases, there will also always be practical difficulties and limitations on how far we can go when environmental, financial, and time constraints are taken into account. So let's start by looking at what we're going to try to achieve. And is this a realistic goal for the farm or the area in question? So if you think we can have no ticks or no tick bites, I think that's unlikely. If you're in an area where there's any rough grazing or possible tick habitats, you're unlikely to get rid of them all. So we should be a bit more realistic than that. Are we looking for elimination of tick-borne diseases from the farm? Again, that's setting the bar quite high, and it does require a lot of efforts on a number of fronts, but it is possible in theory. But we have to reduce the tick population far enough that the tick-borne diseases can no longer circulate. And some diseases are spread through from the adult tick through the egg, so that's very difficult. We do have to look at environmental control, and we also have to look at managing wildlife as well as farmed animals. If we're just after better control of tick-borne diseases, then I think this is always possible. But we do need to look at the costs involved, and it has to be cost effective if we're running a farming business. And again, it will depend on a number of factors, including the farm management, the habitats you've got, the type of vegetation, and the wildlife populations that are wandering around. So let's have a wee look at some tick control tactics. We've really got three things that we can focus on in the overall control plan. The first one is looking at animal treatments. Now this is the easiest and what most people focus on, but on its own it won't give you good long-term tick control. We can look at pasture and habitat management, and we're really talking about reducing tick habitats here and making the environment less suitable for tick survival. And when you look at some of the extensive areas, especially in Scotland, where we've got lots of ticks, it's very difficult to do anything different than we're currently doing with these areas. Grazing management is the third key thing that we have to look at, and we can't just look at farmed animals. We've got to think about the wildlife control as well. So that might be areas where we've got feral goats. It might be areas where you've got lots of deer. But in some way, we have to manage how they interact with the farmed livestock. So let's look at these in a bit more detail. If we look at animal treatments, what we're using is ectoparasiticides. So these are poisonous chemicals, and they work well because they're poisonous and they kill the ticks. We've only really got two groups of chemicals that are available for treating ticks on sheep. The first ones are the synthetic pyrethroids, and these all come as Poron products. There are three different actives in this group. So we've got cypermethrin, alpha cypermethrin, and deltamethrin. And they will give varying levels of tick control from 6 weeks for deltamethrin, 10 weeks for cypermethrin, and anywhere between 8 and 12 for alpha cypermethrin. But you do have to read the instructions on the pack and use them as recommended if you want to get that level of control. So whenever we're using products, we should be thinking about weighing the animals, calibrating our dosing equipment, and reading the instructions if we want to do what it says on the tin. The other option we have is OP dips. Now these have to be plunge dips if we want them to fully soak the sheep and give us the level of protection that we require. And plunge dips will usually give around 6 weeks protection against ticks. There are no products currently licensed for tick control in cattle in the UK. So if ticks are the target parasite, we have to use products that aren't licensed for tick control. And there are ectoparasiticide products licensed for use in cattle, but because this is an off-label use, it should be under veterinary prescription as part of a herd health plan. But if we're grazing cattle in these tick areas, then it's something we've really got to think about. So the chemical treatments are not a quick fix. We really need to be looking at them as part of a broader long-term control strategy. I'll just put up this link here to one of the Moredun animation series. I think it's really important that when we are using products, we use them correctly. And the video resources on the Moredun website are a really good guide to how to get the best out of your medicines. Next on the list will be looking at habitat management. Now, as Mara said, ticks like dense undergrowth that maintains high levels of humidity with some longer vegetation for them to use as a platform to climb or jump onto their host. So these are typical tick habitats here. We've got some rough brackeny ground, we've got some scrubby heathland, and the picture on the right is near where I live, and my dog there was picking up ticks pretty much every month this year. So, you know, there's lots of ticks around, and these areas are very difficult to manage from a tick point of view. So we're really talking about bracken bashing and managing the vegetation that we can where this is possible. It can be effective at controlling bracken and allowing better quality grass to come through. But as anybody who's tried it will know, it's a long-term project and takes time and resources, and you have to keep on top of it. You have to keep at it all the time. And this is fairly typical. We're looking at areas that are steep and often inaccessible, but it's something we do need to think about if we want to have a long-term reduction in tick numbers and better disease control. So it has to be a key part of the plan where it's possible. When we move on to grazing management, we think of two different areas. Firstly, the livestock, where we should have good levels of control. We should be able to catch the animals and treat them when necessary. But we also have to think about why we're grazing animals where we are. And Mara also mentioned this. If we're looking at lambs in endemic areas, we want to be thinking about controlled exposure of these lambs, deliberately exposing them to a low tick challenge so that they can get infected with things like tick-borne fever, tick-borne fever, when it's not going to do them too much harm. So when they're still protected by the cholesterol antibodies from other diseases like louping ill. Because when you get tick-borne fever and louping ill at the same time, because of that immunosuppression, the louping ill is often very severe and often fatal. So avoiding these dual infections or exposure when animals have some level of protection is a good idea. If we're talking about tups, we do know that tick-borne fever will cause high temperatures, and that will cause temporary infertility. So we've either got to get the bought-in animals exposed to ticks and acclimatized well ahead of the breeding season, or keep them, and that needs to be exposed and recovered at least 2 months before before tupping time, or you have to keep them away from any tick exposure until after tupping. We can aim to avoid the highest risk tick areas at times of peak tick activity. And yeah, we used to think of a spring rise and an autumn rise of tick activity, but as I said earlier, my dog was picking up ticks every month this year. So I think that traditional spring and autumn rise has kind of merged into one long sustained period of activity. And that might be down to climate change. It might just be down to the way that we're managing the land. And if there isn't time for controlled exposure, then we need to kind of prioritize the grazing so that our high risk animals are on the lowest risk ground. So lambs that we want to grow fast and finish quickly, bought in animals or pregnant ewes that haven't had prior exposure to ticks need to be protected. And for cattle, there's a nice picture of some hill cows there. There are systems available for virtual fencing. So using GPS collars and virtual mapping, you can then define on an open hill which areas they can and can't graze. And these seem to be quite effective at keeping animals away from various disease challenges. The second part of grazing management is looking at wildlife. And deer can be a problem when it comes to managing ticks. They're big enough to carry large numbers of ticks, and they cover large ranges, so they have the potential to move a lot of ticks and tick-borne diseases from one area to another. In the UK, we have 6 deer species, but the 4 largest species are the ones that are most likely to help the ticks. So we've got roe deer, fallow deer, sika, and red. I haven't got a picture of the sika there, but they look very much like the red deer from a distance. So controlling deer numbers by having an effective culling policy can be effective in areas with lower deer numbers. But as the deer numbers increase, deer fencing is really the only effective option if we want to really keep control over the tick population. We have to think about the smaller wildlife species as well. As Peter was saying earlier on the moorlands, the whole tick control program is often for the benefit of the grouse. But we shouldn't forget things like hares, which tend to go under fences instead of over them like the deer. And we need to think about our dogs as well, because the dogs have the potential to move ticks and tick-borne diseases quite a long way in the back of the car. And again, they are at risk of diseases such as Lyme disease. But keeping things in perspective, the larger the animal and the larger that animal population, the more effective they will be as tick taxis and move the ticks around to places where you don't want them. So if you've got a big deer population, I would focus on that first. So there's quite a bit to think about, but how best do we tackle it? So lots can be done, but what should be done on your farm? And that involves a little bit of risk assessment. So firstly, we need to know what the current disease impacts. And then how much time, effort, and money does this warrant spending on your control program? And then the next question would be, with the budget you've got, what should the priority actions be? And I've put them in this order. So first of all, identify your disease risks and the costs that's impacting on your business. Secondly, identify the tick hotspots. So where are most of the ticks on the farm and where are your lower risk grazing areas? Then we look at animal treatments to limit the disease impacts. So strategic treatments to get the best value for each treatment, depending on the animals that you're treating. And then finally, look at the pasture management, the grazing management, habitat management, and how far can you go with this on the time and budget available. So if we look at those in a little bit more detail, first of all, we need to know what tick diseases are present on the farm. And we'll just run through them again. Mara's covered them, but tick-borne fever, we're mostly looking at sheep. We know it can infect cattle, but it's mostly an issue in sheep. Louping ill, again, mostly affecting the sheep. Babesiosis, mostly affecting cattle. Lyme disease, where we're looking at the shepherds and the sheepdogs. How do we find out? Well, the best way is to test the sheep. So if you've had sheep grazing any moorland area or tick risk area, bringing a number of those in discuss it with your vets and the laboratory or Moredun, and do the send the right number of blood samples in and see what diseases they've been exposed to. It's also worth looking at your flock or herd productivity data and try and get an estimate of what it's costing you in either lost production or lost animals, and then it gives you a better idea of how much you can afford to spend on making it better. Number 2 would be looking at tick hotspots. And Mara showed pictures of people dragging for ticks, and that link there will give you the standard protocol for doing a tick drag. And if you really want to know what the tick population is like, that's the best way of doing it. But if you want a quick and easy one, just count the ticks on untreated sheep. And if we're getting more than 20 ticks on any animal, then I would suggest that would be quite a high risk tick area. Strategic treatments, we've covered this a bit. We've only got those two groups of actives licensed for tick control. And I say tick control products in cattle are off-label, so that has to be part of your veterinary health plan. And we should be trying to use both groups in every treatment cycle to minimize the risk of developing resistance in the tick population to either one. Now, I know we don't have any resistance detected so far, but that doesn't mean we can sit back, because if we're not careful, it will happen. Other parts of the world, they have massive tick problems and huge levels of resistance to all the actives that they have available. So we should count ourselves lucky and look after the products that we've got. We have to manage the wildlife tick taxis if we were aiming to reduce the tick population and reduce tick diseases by doing that. And when to treat depends entirely on your flock or herd management and the areas being grazed. So this is absolutely specific to your farm. And then habitat management and wildlife comes down to rush control and bracken bashing. And this little tractor is really quite useful for the gentle hills and lowland areas. But if we're talking about steep, inaccessible areas, then you do require some pretty specialist equipment. We need to avoid certain areas for high-risk animals at the high-risk times of year. And we need to think about maybe using virtual fencing if we've got cattle that are grazing these risk areas. And then either a population control policy or some deer fencing if deer are an issue. And what to do and when on this? Well, it depends on what you hope to achieve, but also on the budget and the specific area that you're trying to manage. So again, this is absolutely specific to your farm. So in summary, there's lots to think about. There's a lot that can be done, but the plan has to be specific to your farm. So it's worth sitting down with an advisor who understands tick diseases and the treatment and management options available, and working out a plan that you can manage with the time that you have and the budget that you have. So thank you for your attention, and I'll hand you back to Peter now for any questions.
Thank you very much, Matthew. There's a lot of food for thought there. I will have questions now, but I would quite like to ask Mara first, just to get things going. You talked about developing a new vaccine. There was a louping ill vaccine that we used with the gamekeepers some years ago. It was a horrible vaccine to use. It was really quite difficult to use, very thick. It was like Copydex. And then it came off the market because it didn't justify the manufacturing cost. But the one thing I did notice, I discovered using the vaccine, was that you had to keep the animals away from the tick-infested areas for a number of days before you exposed them after the vaccine, because they had to develop immunity. So we found to our cost that vaccinating animals and then exposing them to the tick-borne immediately, I suddenly had a heap of dead ones. So I would warn on that. But I'm interested to know if there's a new vaccine coming out, Mara.
Yeah, so the new vaccine, it's been developed. The first, as you know, a vaccine needs to go through a set of development and trials before it can be put forward for authorization and marketing. So we have completed the first vaccine trial at the end of May, but they're still analyzing the results. And the second vaccine trial is underway just now. And the reason why we have to do a repeated number of vaccine trials before we can really analyze the results is that because Laupin-Gill, it's a disease that it's zoonotic, can give a neuroencephalitis in human, and we have to do it in specific facilities where we can only host a certain number of animals at the time. So we can't do a big group of animals all at once. We have to do a number of small animals and repeat the test. But I spoke to the scientist that is doing this work, and I think there won't be, not before 2 years, before the vaccine, if everything goes well, is allowed to come to the market.
Thank you, Mara. We have a question here from Andrew Forbes. If we had effective vaccines for the most common tick-borne diseases, do ticks need to be controlled? As I can't read that. Ricinus typically causes relatively little direct damage.
Yeah, the tick itself, unless it's a very heavy infestation and gives anemia to your animals, it doesn't cause particularly heavy damage. Although most of those vaccines are going to be very difficult, especially the one against tick-borne fever. The attempt to produce a vaccine against tick-borne fever goes back to the early 50s, but the way in which the bacteria behaves keep changing what it shows to the immune system during the development of the disease, and the fact that it affects directly the immune cells that are the ones that are supposed to combat it, have resulted in a number of vaccination trials and in a very large number of negative results. And I don't think is there anybody at the moment that is trying to develop that type of disease. It will require a completely different way of thinking, a completely different approach, and probably a large investment from a monetary point of view.
Yeah, I would agree with Mara on that. The diseases that we're at the current time, we're not going to have vaccines for them. But if you're looking at managing ticks for grouse populations, then even without the diseases, a small number of ticks can cause severe anemia in grouse chicks, and so we have to control numbers for that. You're still on mute, Peter.
I was on mute now, I can't find the blinking question. Here we go. For Matthew, how would you expose lambs to tick-borne fever without the ewes getting it? For Matthew.
Well, the thing there is that we're probably looking at ewes that are adapted to the hill environment anyway. They've been resident in that area for a while, so they will have met ticks and tick-borne diseases before they lamb. So we're talking about hefted flocks mostly. And the aim there is to make sure the lambs go back to the hill within the first 12 weeks of life, when they've still got maternal antibodies circulating, so they can get a few tick bites and hopefully get over tick-borne fever before their borrowed immunity to Laupin-Gill wears off.
Here's another question. Is dipping a good option for tick mops, or does the dip deter ticks from jumping on a host?
I think dipping is a very good option. Both of these chemical groups work because they're poisonous. So as soon as the tick jumps on, it will be killed, which is what you want for your tick mops. but again, we've got to be careful how we use them. So follow the instructions. Don't allow these animals to have access to water courses, and yeah, mix it up a bit. Don't use the same group, the same chemical too often in a row.
I know personally that we dip twice a year, and we use the cypermethrin drugs, Porons, 3 times a year. So we mix and match. The Porons last considerably longer. A thing that I noticed with the dissect, zermisect chemical, make sure and keep your dogs well away. I've seen the dogs eating away at bits of broken wool that's had the chemical poured on it, and the dogs ended up in the vet. So make sure and keep your dogs well clear of these chemicals, because there can be quite drastic effects on them.
Yeah.
Have we any more questions? Well, we'll have some questions here. Should we be treating lambs before they go to the hill, or should we leave them for the first cycle to build up immunity? That's a good one.
It depends on the level of tick challenge. So I know certainly in the hill areas on the west coast of Scotland, where there are an awful lot of ticks, you need to give them some, a little bit of spot on as soon as you can catch them. Even though it's a shorter acting one, the deltamethrin is okay to put on newborn lambs. And if you do that, then they get a few tick bites as it wears off, rather than getting eaten alive as soon as they go back to the hill.
Thank you. And here's one from Andrew Forbes. Given some of the successful use of cattle breeding for tick-borne diseases, resistance, resilience and control in the tropics, are there any prospects of the UK for utilising native breeds of cattle or sheep?
I think there's always potential, but I don't know enough about redwater and babesiosis to know if it's an inherited ability to cope with the disease, or if it's just the luck of the draw, whether you get a severe infection and die, or a mild infection and survive. Mara, do you know anything more about those?
No, not really related to babesia. What I know is that it's a specific breed that are the ndama that in the tropics show a resistance, but it's not a resistance to ticks, it's a resistance to specific diseases. So again, you can try breeding, but there is always trade-off when you try to enhance a breed in one way or to the other. So you need to keep in mind that, first of all, you have to demonstrate that it is actually factual, it's not just anecdotal. And then you might need to understand what else you're trading off when you're trying to breed for this trait.
Mm-hmm.
Okay, thank you for that. Another question here. Does the chemicals in Porons go into the bloodstream? You know, is that how it's circulated around it, so that the ticks get killed? Is it in the bloodstream?
No, most of the poron and dips as well sit on the skin and are bound into the lanolin, into the wool grease. There's only a very small amount that gets into the animal and excreted through the system. So mostly it's on the surface and is bound into the lanolin. So as soon as the tick jumps on, it gets some exposure. The longer it's been on, the less there is present. So, with the porons, you'll often get ticks biting before they die. It's just they need longer to absorb enough chemical to kill them.
Okay, another one here. Are there any new poron treatments being developed at the moment?
No. I think that's quite an easy one. These are poisonous chemicals, and there's nothing new in development as far as I know along the ectoparasiticide line.
Hence the reason it's really important to mix and match so that you don't get resistant to any of the chemicals.
Yeah.
Okay, here's another one. Do natural England grasp seriousness of ticks? We have experienced a tick boom in the Peak District, having not had the issue before, and Northeast seem oblivious. or Natural England, sorry, NE, oblivious and don't seem to be willing to look at increasing sheep numbers and assisting us to get permission or for fencing in the NP. Do Moredun have any involvement with Natural England? Beth, maybe you can tell us.
Yeah, hi folks. Not that I know of. No, we do work with some of the national parks, but we haven't had any dealings with Natural England, but that's not to say that we couldn't. So we'd be pretty keen to speak to them. So if you've got an issue and you want us to speak to MD from Natural England about ticks and try, maybe they're on the webinar actually, or try and educate them, we're very happy to do so. We've also, as Mara brought up, got some really good resources for ticks that we could send out to them. So, you know, if you want to get in touch with us afterwards, we're quite happy to do that.
Thank you, Beth. I'll just explain to the people listening. Beth Wells is with the Moredun Institute and she's assisting with us today, and that's why I asked her. Here's another question here. There's a tablet against ticks for dogs. Is there anything similar for livestock?
There isn't at the moment. That's a different group of chemicals, and at the moment they're not available or likely to be licensed for use in livestock in the UK because of the way they work. These are systemic products, so they're absorbed into the system, and for cattle and sheep that would give them an extremely long withdrawal period. So you would then be struggling to market these animals for a long time. So I think the chances of having these as an oral product are very slim. Obviously, people are looking at whether these are going to be useful for tick products in the UK, but like I said, it's unlikely to come because a lot of them are in the kind of neonicotinoid group, which is something that you know, has been banned in crop chemicals. So I think we're unlikely to get it through as a treatment for animals. Okay,
thank you. Has there been any resistance to Porons solutions been found in the UK yet? I think there has in my neck of the woods.
It's a question for Beth, because there is a person that is currently doing a master looking at what are at the moment anecdotally report resistance to tick treatment. I don't know if Beth want to say more.
Beth, do you want to come in?
Yeah, thanks, Peter and Mara. Yeah, there is some work going on at the moment. There's a questionnaire that's actually still live, I think, till about the 11th of this month. And the links are actually on the Moredun website. So if you want to go and take part, that's really good. We'll hear probably towards the end of the year what her outcomes are. But I think I'm probably right in saying, Matt's going to correct me if I'm wrong, that so far there's been no actual resistance found to any of the Poron solutions.
Well, that's right. There's been nothing confirmed in the UK. But if you think you're having problems with products not doing what you think they should, then please do report it to the manufacturers, because they'll always be interested in finding out what's causing it. And it really is important that if you think the product hasn't done what it should, that it gets investigated, because if we're starting to see resistance, then we do need to know about it. I'll just say, in most of these cases, it's down to either not weighing the animals and just making a poor guess at their actual weight, so the animal doesn't get enough of the product, or the product maybe doesn't get used in the correct way, so it doesn't get onto the bits of the animal that it needs to. So there are other reasons for treatment failures other than resistance, and we do see some of that every year. So if you think you've had a treatment that has failed to do the job, let the manufacturer know and they'll investigate it and hopefully help you avoid it in the future.
Thank you, Matthew. We have another one here from the Game Conservancy. Had some success trialling dog collars with deltamethrin in North Yorkshire. Have you heard about this one?
dog collars with deltamethrin. I did hear about it, but I'm not sure if that product is still available, because the current dog collars have a different active in them, which again is probably not one that would be available for sheep or cattle.
Okay, and another one here. How often do you need to change Porons, i.e. Monthly or yearly?
So all the Porons are the same chemical group. So changing between a deltamethrin and a cypermethrin or an alpha cypermethrin isn't a change. It's the same chemical group, they work in the same way. And if you start to develop resistance to one, you've developed resistance to them all. So the only change you can do is between a Poron and a dip.
Excellent, excellent. I don't see any more questions here. Do we have any more questions? Let's come into the end of our time here anyway. If we've no more questions, I would like to thank most sincerely our panelists tonight, Matt Colston and Mara Rocchi. Thank you very, very much for a most informative presentation about ticks, and Beth there in the background keeping us recht. Thank you very much. It's a subject very close to my heart because I've been dealing with it for quite some time, and it's great to hear the experts who know what they're talking about. So on behalf of everyone here tonight, thank you very much to our panelists, and thank you very much to the NSA and for the Moredun for putting on such an informative and helpful topic. Thank you very much, and good night.
Thank you, Peter.